Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump in, please do hit that like button and subscribe. It really helps support the Sand Science Experience, helps us keep bringing you these deep dives. So today we're digging into something pretty fascinating. It's right at that intersection of AI and biology, a system called Crispr-Gpt. Our mission here is to sort of pull back the curtain on how this AI co-pilot is using large language models, LLMs, to really change the game for gene editing experiments. And our source for this is a brand new article just published in Nature Biomedical Engineering.
That's right. And CRISPR-Gpt, it's not just, another LLM off the shelf. It's what we call an LLM agent system. That means it can reason, it can plan, and it can even help execute tasks. Think of it like a digital research partner. What makes it really stand out, though, is how it blends the general smarts of an LLM with very specific, deep biological knowledge. And it gets this knowledge not just from papers, but by using retrieval, plugging into external tools, and even, this is quite unique, it's fine-tuned on thousands of real discussions between scientists on open forums.
Okay, let's unpack this. So it's like having an expert biologist and a super smart AI assistant rolled into one, but one that's learned the ropes from actual scientists talking shop. Practically, if I'm in the lab, what's it like using this thing?
Exactly. That's a great way to put it. It actually offers 3 different modes depending on what you need. First, there's meta mode. Let's say you're totally new to gene editing. This mode gives you step-by-step guidance, everything from picking the right CRISPR system, those molecular scissors we use, to designing the guide RNAs, which are like the GPS coordinates for editing, and even drafting the whole lab protocol. Then you've got auto mode. This is more for experienced researchers. You can give it a sort of freestyle request, a high-level goal, and the system itself figures out the steps, breaks it down into a custom workflow, manages how the tasks depend on each other. Pretty smart. And finally, there's Q&A mode. This is like having an instant expert. You can ask anything. It pulls answers together from papers, from established protocols, and importantly, from this specialized model called CRISPR-Llama 3. That's the one fine-tuned on over 4,000 scientific discussion threads. So it really has that kind of scientist's intuition baked in. It understands the nuances.
Right, that scientist's brain part sounds, well, frankly, revolutionary. But, does it work? Is there proof this actually delivers in a real lab?
Yes, the validation.
Yeah.
And the results are pretty striking. The paper talks about actual wet lab experiments. Get this. Junior researchers, people without prior gene editing experience, used CRISPR-Gpt to design complex experiments. And they succeeded on their first try.
First try, seriously?
Seriously. For example, they successfully knocked out 4 different genes like TGFR1 and BEX involved in cell processes in human lung cancer cells using CRISPR-Cas12a, which is a specific type of CRISPR. And another experiment, they epigenetically activated 2 genes, NCR3LG1 and CACAM1 in melanoma cells using a different tool, CRISPR-Cas9. So switching genes off and on.
That's incredible. So it's not just theory. These are real, tangible results. How exactly does it take them from just an idea to, getting those results? What's it doing behind the scenes?
Right, and that's the key to why it gets those results. It handles the entire experimental process end to end. We're not just talking about one piece. It helps select the best CRISPR system for that specific job. It designs the guide RNAs, even suggesting the best spots, like the optimal exons, within a gene to target. It helps choose how to get the CRISPR tools into the cells, drafts those super detailed protocols we talked about, and then after the experiment, it helps analyze the data using built-in tools like Primer3 or CRISPR-SO2 to check if the edits actually worked and how well.
Wow.
Imagine the time saved, all the different bits of expertise you'd normally need. CRISPR-Gpt brings it all together.
So it sounds like it's not just making things faster, it's making gene editing more accessible, lowering the barrier to entry, which is huge. So what does this all mean for the pace of scientific discovery? This could really accelerate things, couldn't it?
Absolutely. It speeds things up, reduces errors, improves reproducibility, all by making this powerful tech easier to use correctly. But this raises an important question. With such a powerful tool, especially in gene editing, we have to talk about ethics and safety.
Yes.
Definitely.
That's critical. What guardrails are in place?
The developers seem to have thought carefully about this. CRISPR-Gpt has several built-in safety layers. If you try to do anything involving editing human tissues or organs directly, it flags it. It gives warning notes, provides links to the international rules about heritable genome editing, the kind that gets passed down generations, and you have to actively confirm you've seen them. Crucially, it's designed to identify and just stop requests that involve editing human germline cells, sperm or eggs, or requests involving dangerous viruses. It just won't proceed. And for data privacy, which is another big one, it It blocks long DNA sequences, anything 20 bases or more that could potentially identify someone from being sent out to external LLMs. It prompts you to remove that kind of sensitive info first.
Okay, so it sounds like it's built not just for power and efficiency, but with responsible use and ethical considerations baked right in from the start. That's reassuring. Well, this deep dive into CRISPR-Gpt really highlights the incredible potential here. AI systems as genuine co-pilots in the lab. Just imagine how much faster we could unravel complex biology, maybe find new cures, when AI can help guide almost every step of an experiment. Makes you wonder, what new frontiers will this open up for you to explore? Thanks for joining us. Don't forget to like this video and subscribe to the Science Experience podcast for more deep dives into the cutting edge of science.